In our preceding lectures, we've talked about how to do good peak listing. We've talked now about how to do retention time mapping and feature mapping across experiments. But before we start talking about how to harvest information from these reports, I think it'll be really valuable if we spend some time talking about statistics. So when we talk about differentiation in XCMS, we're joining ourselves to a long tradition of many different strategies for finding differences in biological samples. So we'll start by just talking about difference testing generically, specifically for the challenges of biological mass spectrometry. And from there, we're going to branch off into some of the statistical designs that are possible when we make use of XCMS. Now, a lot of the things we talk about with XCMS are features that are available within the XCMS online interface, which may not be as accessible from some of the things that we've done here. But the principles should be uniform throughout. So a lot of times when we talk about the capabilities of mass spectrometry, we're talking about we're caught up in the bind that we're very, very good at positioning a peak in M over Z space. But our ability to measure intensity directly is not quite as solid. The degree of variation that we experience can be kind of high. So when we work with a Fourier transform mass spectrometer, we generally expect our masses to be accurate within 10 ppm. I mean, that's like more than 2 standard deviations away from what we expected those instruments to produce. When we deal with intensity measurement, we're almost always dealing with a far greater degree of variability. And that is something that we have to take into account when we're dealing with difference testing. So when we look at very well-tuned quantitative experiments from triple quadrupole mass analyzers, we're typically seeing a 10% coefficient of variation as kind of a starting point. If your measurement technology is achieving at best a 40% CV, it doesn't really deserve the name quantitative at that point. So one of the things that I always try to remind people is that if you are doing a shotgun experiment, if you're doing a wide open discovery experiment, there are going to be limitations on the quantitative precision that you can achieve. These experiments that we've been working with, the Coke versus Pepsi example and so on, they are not designed as targeted quantitation experiments. They're wide open. So any ion that happens to fall on the right retention time and M over Z space will be considered as a possible difference. If you want to quantify those differences more accurately, it's really important that you design an experiment around that. So doing a targeted experiment is far better in terms of the precision that we see in the numbers that come out. These are figures that were produced in the original Max Quant paper, and I think that they are useful for us to think about. When we look at a given peak, that peak has a 3-dimensional representation. We have the M over Z values that any of the intensity fall within. We have the retention time through which we're seeing those, and we have intensity patterns that we hope will be nice and distinct, right? I mean, we're all hoping for data that are this lovely. They've got this nice rounded peak here, a limited amount of tailing to substantiate it, and you don't see a whole bunch of jaggedness and bouncing up and down in the intensity signal. Of course, as you get to lower and lower intensities, though, the degree of variation that we see in intensity is going to grow. So, being able to deal with these as quantitative data requires that we have some sensitivity to that amount of variation. And as we talk about variation, we should also be thinking about the expectations of the statistical tests that we're using to make sense of those data. Now, we frequently hear the terms parametric and non-parametric when we're dealing with a statistician, and some of us might understand what those terms mean, but it's really better for all of us that we understand what those terms mean as we go along, because these have everything to do with what kind of assumptions are built into the statistical models that we're trying to apply to our data. So when we talk about things like students' t-test, we're talking about a parametric model. And that is to say that it has some prior expectations about the data when you're applying it. Which is to say, it expects those data to come from a specified distribution. specifically the Gaussian or normal distribution. So if the variation that we see in our values are not Gaussian, using a t-test is going to produce skewed p-values. That's not okay. So if you want an accurate estimation, make sure that the test you're applying actually has something to do with the data, the underlying data that you're working with. Non-parametric tests can be quite good, and even better, most of them don't come with as many expectations or assumptions about the underlying data as do parametric tests. So lots of people have heard of the t-test. Has anyone in this room not heard of t-test? Okay. T-test is very well known, and that is good. The U-test is its corresponding partner, kind of, in the non-parametric space. So the MannWhitney or Wilcoxon test, it's kind of been put together in a few different ways, is very useful when you don't really know much about the distributional expectations of your data sets. Generally speaking, for a reasonable number of replicates, the p-values that you get from non-parametric tests are just as good as the ones that you get from parametric tests. But there are some limitations to these, and we'll talk about those as we move ahead. So in this case, I have created a model of two distributions. Imagine a world in which we were able to get 20 replicates, right? That would be brilliant. So 20 replicates versus 20 replicates. Here I have told the software, this is R being used in this case actually, we are grabbing out two distributions. We're sampling 20 peaks from two different distributions, one with a mean of 3 and one with a mean of 4, with a standard deviation of 1. All right, quick reminder, what's a standard deviation mean in the context of a normal distribution?
One standard deviation to each side from the center, that area would be 2/3 of the whole area.
Yeah, that's good. All right, so for a first approximation, a standard deviation tells you is it fat or is it skinny, right? In this case, you're correct. Yes, when we're moving, when we allow ourselves to move up one standard deviation from the mean or down one standard deviation from the mean, we're usually accounting for, I believe, about 68% of the total distribution. Yeah, so yeah, 2/3 is a great estimate for that. If you have a standard deviation of, if you're talking about going up by 2 standard deviations or down by 2 standard deviations, you account for about 95% of the distribution. And if you step 3 out, you've got about 99% of the data set covered. So when we have a standard deviation of 1 and a mean difference of 1, our ability to tell these two distributions apart is somewhat limited. I mean, these overlap quite significantly, don't they? When we compare the 20 values drawn from the two distributions, we get a significant p-value, whether we're doing it by t-test or by the Wilcoxon-MannWhitney approach. Both of them give us significant p-values in that case. So that's great. But what I want you to think about in your mind is how confident you can be that the differences are significant if these standard deviations were 2 instead of 1. What if the means were different by 0.5 rather than 1? What kind of impact would that have? So what you should keep in mind then is that we're comparing two different effects. One is how different are the means, and the other is how variable are the distributions around those means. Both of those factor into whether this p-value that comes out is significant or not. All right. So in the case of, in this case, both tests give us significant p-values. The student's t-test has a pretty long history, actually. Do we have any fans of Guinness in the room? No beer drinkers? We have a couple beer drinkers? Okay. Oh yeah, we got a Guinness back there, okay. All right, so Guinness had a dirty little secret back in the early 1900s. They had hired a statistician because they thought that a statistician might help them to understand better how to keep their beer making in well-controlled processes, so that they would always get a good batch of beer at the end. So they'd hired a statistician, and the statistician named Gossett put together an interesting little test for comparing the means of two distributions. So he thought that was pretty noteworthy and thought maybe other people would like to know about it. But Guinness wouldn't let him publish in his own name. Instead, he had to publish under the name student. And so when you write about student's t-test, you should always capitalize the S, because student is his pen name in this case. All right. So what we're trying to do then is evaluate the difference in means based on taking into account the variance of the samples or the standard deviations of the samples. That produces a t-statistic, and that t-statistic can be looked up to produce a p-value. So when you get a p-value, the p-value does not go low to tell you that your hypothesis that they're different is correct. Okay? I'm sure we all heard that once at least in our college educations, but let's re-emphasize that. The p-value does not enable you to accept your alternative hypothesis. What it does give you the ability to do is discard your null hypothesis. The null hypothesis is that there's no difference between these two distributions. So we want to evaluate the probability that by random chance the means would look this different given these underlying data. All right? So when we get a p-value out, it tells us whether or not what we're seeing is likely to have happened by random chance. However, it assumes normal distributions in the data that we've seen. Now, normal distributions are a pretty good bet in most cases, because when we have lots of factors that are contributing to variability, we assume kind of a normality in the result that comes out of it. But there are certainly some cases in which the variation that we see in these intensities are not normal. And if you have a case like that, you can't do a t-test against them, because that will give you a misestimated p-value. Now, MannWhitney Wilcoxon is rather different. Instead, what we have are, imagine our 20 data points. We had 20 data points for A and 20 data points for B in those curves that we were looking at just a minute ago. Imagine that we have them arranged on a number line from lowest value to highest value. And we have a red marble to indicate every place where we had a value from A, and we use a blue marble for every place where we had a value from B. So if we imagine what this looks like, we could have a scenario where every A is higher than every B. All right? So we'd have a bunch of red marbles here, and we'd have a bunch of blue marbles there, and all of the reds would be past all of the blues. That'd be great. So that corresponds to the most extreme p-value statistic that you could get out of a MannWhitney Wilcoxon test. If you have some mixing in the red and blue marbles, though, you might ask, how likely is this particular interdigitation of red and blue marbles to be by random chance alone? And basically, the more mixing you've got between the distributions, the more that your p-value is going to range upward and upward. So, does it matter in a MannWhitney test if all of the reds are way over here and all of the blues are way over there? Not really. That gives you exactly the same p-value as if they're right next to each other. It's the mixing that leads to that p-value going up. So, as we move forward, you should keep in mind that difference between a parametric and a non-parametric assessment of differences in these two distributions. Now, I've written in a caution down here at the bottom. Don't use this in a 3x3 test. Why is that? 3x3. All right. So imagine that I have 3 red marbles and 3 blue marbles. What's the random chance? The random chance that all of these are above all of those. Let's make it even simpler. Let's say that I've got only 2 replicates of each. Okay, so now I've got 4 values. They could be all the reds above all the blues. They could be all the blues above all the reds. They could be mingled in a couple different ways. But as you see, the number of possible permutations of these marbles is pretty small. As a result, the most significant p-value you can get with all of the reds above all of the blues, or vice versa, can happen very frequently by random chance. As a result, the most significant test you can get out of a 2 by 2 is not very significant at all. I mean, it's never going to get below the 0.05 value. And the same is true for a 3 by 3, because even though there are more combinations by which you can squeeze red and blue marbles among each other when you've got 3 of each, the most significant value that you can achieve is just not that, it's not that low a p-value. So I would really suggest that you have at least 5 versus 5 if you're going to make use of a non-parametric test like this. All right. That brings us to the problem of the curse of dimensionality. We speak of the curse of dimensionality frequently in systems biology, and the reason for that is that we have many, many tests that we can perform because we're measuring lots of things, but generally with very few replicates. Now we see that the scale at which people are practicing systems biology is always increasing. The number of samples that we see in tests is no longer, say, 3 versus 3. We frequently see, even more than 100 samples being thrown together in some of the big systems biology profiles. That helps. But I would also note that the number of entities that we're measuring in our experiments is always going up. So let us imagine that I have 20 different ions in my metabolomic experiment. And for each, I have some amount of intensity seen in my 5 versus 5 experiment. So 5 replicates of A, 5 replicates of B, and I have a table of 20 different species that I'm evaluating. So I might then compute a p-value, let's say by the t-test, for each of these 20 items. Okay, so what distribution do I expect the p-values to follow if there's no difference?
Yes.
Should be mostly uniform, actually. We expect a uniform distribution of these values. So by random chance alone, how many of these 20 tests that I've produced should produce a significant p-value of below 0.05? How many p-values did I compute? 20, okay. So I computed 20 p-values. What number of those would I expect by random chance alone to have a p-value below 0.05? If they're uniformly distributed over the range 0 to 1, 1. By random chance alone, no difference at all between the two cohorts, I expect one of those 20 metabolites to be significant. So that's a problem. Because frequently, we're not dealing with 20 metabolites, we're dealing with 20,000 metabolites, or 20,000 genes, or 20,000 proteins. So in cases like that, our ability to find significance has this little problem to it, that we have far more tests that we're performing than we have replicates to keep those values down. Keep them up, actually. So there have been 2 major approaches that have fallen out for dealing with these. I'm also going to talk about a third as well. So let's start with the most conservative of them. Bonferroni correction is one of the ways that we frequently see in Manhattan plots for GWAS, genome-wide association studies. The notion behind this is that if you are running 20 tests, you should be 20 times as stringent in accepting them. Okay? So if your standard for applying a p-value threshold typically is 0.05, and you're performing 20 tests, you divide 0.05 by 20 to get your new threshold. So when you're looking at a Manhattan plot from a GWAS, from a genome-wide association study, you'll frequently see a line drawn across that represents the Bonferroni corrected p-value threshold. So Bonferroni is strict, and that is a really, really hard criterion to beat. Certainly having lots of replicates and very low variability matters a lot in trying to beat a Bonferroni corrected p-value test. The next route is the Benjamini-Hochberg approach. More frequently we hear it called the family-wide false discovery rate. So in this approach, we're using a different kind of correction to these p-values to reflect ever greater stringency as we've performed more of these tests. But there are some other tricks that we can use. Let's drop back to our 5 red versus 5 cohort A versus 5 cohort B, 20 metabolites. There's another approach that we can use to figure out what the random background model of what these p-values should look like. And that's simply to mix them up. Originally, we had these 5 being all cohort A, and these 5 were all cohort B. But we can scramble them up and relabel them to say these are now A, these are now B, and see what the p-value distributions look like when we've got them jumbled up like this. So that gives you another way to correct what these p-values are, to evaluate in kind of a randomly shuffled distribution instead of using the Benjamin E. Hochberg Bonferroni approaches. All right. So when we work with XCMS, we see a variety of comparisons that come into play. Now, I've spent an awful lot of time talking about 2 cohort comparisons. These are these A versus B comparisons I keep talking about. So you can see from this recent publication, this is from 2014. I think that this publication is on your hard drive already from the thumb drive today. But if you want to compare 2 independent groups, you can do an independent t-test. This is the Welch test. So Welch made a minor fix to the student t-test that allows for the possibility that the A's and the B's will have a different variance for each set. So the independent t-test gets used for that. Now you may have 2 dependent groups. You may have 2 different time points, for example, for each of 5 patients. So for something like that, you have 2 dependent groups, and for that, a paired t-test is appropriate. Now, it may be that you want to make use of this, the fewer assumptions of a non-parametric test. And so they've got the MannWhitney or Wilcoxon test in here. The Wilcoxon is the signed rank test. They use this for evaluating basically how much the rank changes from the early to the late. And they also have a fair number of visualization tools built around that. They also have the ability to do comparison of multiple independent groups. You can do that by ANOVA. If you want to do the ANOVA, except this time without the assumptions about how it's parameterized, you can do a Krustal-Wallace test instead. And they have related sets, sort of the equivalent of a paired test in the multivariate space as well. So, a lot of people don't really think about the difference between a paired t-test and a standard t-test, and you really need to pay attention to that because using a non-paired t-test on paired data can lead to really weird results. So I would note that if you were to look at all of the before measurements, you get some sort of distribution out, and if you look at all of the after measurements, you get some sort of distribution that falls out of that. But if you do this as a paired t-test, you're going to get a very different result. And the reason is that there's a fixed value differentiating all of the befores from all of the afters. So the after result is very heavily dependent on that because the low values we see in after are also low values in the before. So the p-values that pop out of that are going to be quite different. So nothing else I really care to say there, so let's move on. Volcano plots are becoming very frequently seen across systems biology, and understanding them, I think, is very valuable as well. So in this case, we have a fold change. So some metabolite has maybe a low concentration in the A's and a high concentration in the B's. So that's going to lead to a big fold change. Typically, we show these on a log scale. The reason for this is to flatten out these differences. If you have a 10-to-1 ratio in something, it looks much different than 5-to-1 if you do them on a linear scale. But if you're doing them in log scale, that big distance kind of flattens out. Essentially, you can think of a log fold change as being as being a way to flatten out all that distance. So as we look at negative log 10 p-values, again we're flattening out this distribution. So when we have a significant p-value, it's pretty close to 0 if we look at it on a linear scale. But if we throw it into negative log 10 space, suddenly those values go high. So a high negative log p-value is much closer to significance. And in this case, we see that we can separate this into in a couple of different ways. So just throwing myself in front of it, anything that's really close to the 0 line in the middle is going to be not a very big fold change. And generally speaking in biology, if we see a very narrow, a very small log fold change, even if it's significant, we tend to ignore it. So anything close to 0 on here is going to be called not a big enough change to care about. Next, we have another line that we've thrown in here, and anything above it and outside the middle is going to be colored blue. So in this case, it's using it, the blue sector here is to show things that have a fold change that's big enough to be meaningful, meaning they're far away from the middle, and they're high enough to be significant. So whenever you're reading these plots, it's always worthwhile to keep in mind those kind of two lanes. You're looking for things that are outside the middle and things that are high enough to be called significant. All right. Now they've gone with some really novel ways to do the visualization. So not volcano plots, but rather these cloud plots on the XCMS interactive site. And I would note that looking at these on the website itself is quite valuable because you can see just how much information becomes accessible to you this way. They're much more interesting to look at in that light. So we start with a total ion chromatogram. So we're looking at how much intensity is seen as a function of time across here. And then we're also looking at M over Z values. So these M over Z values are represented by where vertically these dots are drawn. So one that's drawn here has was observed in negative polarity around negative 600. The color is going to be associated with the p-value. So the lighter it is, the higher the p-value, right? I think it's actually the higher the negative log p-value is. And the larger the radius, the bigger the change. So it's kind of the same information as the volcano plot, but they're showing a lot more behind it. And you also get some sense of where the most ions were seen. There's some really strong signals in here, for example, that differentiate this. All right, so you have a lot of options to modify how this is visualized as well within the user interface. It's very valuable. Now, we're not going to be producing any of those in the hands-on session because those are not what we're generating. We don't have the cloud plot software installed here. But these are all accessible if you've done your workup within the XCMS online site. All right. Now, meta-analysis is a huge and somewhat controversial area, actually, because there are lots of ways in which trying to combine information from multiple studies can get you in a lot of trouble. Meta-analysis within XCMS has given you quite a few ways to go. So let's start with this statement of the problem. My experiment produced 147 putative differences. Right? So I have significant p-values for 147 things. But people who've studied this system before produced 124 and 78 differences, respectively. So my difference list is longer than theirs. Does that mean I should celebrate? Maybe. Maybe you should. Maybe you've got more replicates built into your study. Maybe you have more sensitive mass spectrometers, and so on. So it could be that the longer list of differences has to do with a better experiment. That is possible. But that is not the only possible explanation. You may have variability that's crept into your experimental design that causes things to look different even when they're not. So being able to show how your study overlaps with prior studies is a pretty big deal. So there are lots of Venn diagram possible ways to examine these results. So if, for example, your 147 results are a superset of the 124 and 78, if those are all contained within your experiment, that's really about the best outcome that could plausibly happen. You've got better sensitivity, and all of the prior results have been confirmed by yours. What if a much smaller fraction, however, are mapped? What if only half of the 124 were seen in your experiment as well? Maybe we feel less enthusiastic about that. So this is the kind of thing that we can do using XCMS Online. Also, there's a very handy tool called Venni. I don't know if you've had a chance to see it, but it seems like when I see posters at conferences, 9 out of 10 Venn diagrams have been produced by it. It's a very useful site, and I would encourage anybody who hasn't worked with it to use it. For example, if you put in the names, David, Charlie, and George, and then put in Anita and Charlie and George, the software will figure out what the overlaps are for you. So it's really quite handy. Okay. So if you have a multi-group comparison, you might imagine, we'll call them A, B, and C, that one way to deal with what's different among these experiments is to compare the A's to the B's, compare the B's to the C's, and then compare the A's to the A's to the C's. But there's a weakness in that because you'll have computed a whole lot more tests than was actually appropriate to do. Doing it once in an ANOVA test gives you the ability to take these different factors into account. So instead of doing a more pairwise test than you actually have to do, the software will be able to take into account kind of the was seen in A, was seen in B, was seen in C in one swoop instead. It may be that many different metabolites are all signals of the same thing, right? So what if, for example, all the PCs, the phosphatidylcholine lipids in a sample are happening in concert? What if they are all rising together? To treat each as an independent signal would be kind of a mistake. So you can use principal components analysis to recognize the covariance of these lipids together. So as they covary, they can be grouped together in a single component that you can use to explain your results a lot more compactly. All right. So I'm showing slides of these, and here we go. So this is the ANOVA example. So we have a sample type that can contribute. So we've got 5 different samples that can come into play. And we can look at individual metabolites, in this case glutamic acid, to ask how do the measurements of this particular metabolite vary across these. And we can see that we're getting p-values out of these. Now this is kind of a half a cloud plot, basically. So we can evaluate which ones have p-values, and by that, bull this out. So this is an interactive chart. By clicking on one of these globes down here, I'm going to get that particular metabolite's raw data represented in this kind of way. All right. So here you can see that you can set what are the critical thresholds for different features. And it may be that your interest lies only within a narrow M/Z range or within a narrow retention time range. So having seen a blob show up in all of the A's but none of the B's might draw your attention to that particular area. You can narrow down that to figure out what exactly that looks like. Tennis elbow? Oh, okay. So here we go. So now we're looking at a principal components analysis of these data sets. Keep in mind what I was saying yesterday, that when you're looking at a principal components plot, you're almost always looking at just the first 2 components. Now, by definition, those first 2 components are the ones that account for the most variance in the experiment. So generally speaking, that's a good approximation. But you may yet have greater variation showing up, or additional variation being accounted for by the 3rd principal component. So in this case, we're working over, I believe, the same data set, and we're looking at how these different experiments play out over principal component space. Loadings give us some ability to ask how much individual ions are contributing to it. So you may want to know, you may have observed that principal component 1 is very good for separating samples of this class from samples of that class. And now you want to know which metabolites are contributing to this separation. So you might look at the loadings for principal component 1 to figure that out. All right, which brings us to our takeaway slide. XCMS is certainly capable of much more complex designs than simple cohort A versus cohort B designs. And doing that means moving to statistical tests that I think are a little less comfortable for a lot of people. I always find it rather hard to to conceptualize ANOVA in explaining it. I mean, using it is one thing, but understanding it, I think, is quite another. Certainly, people feel much more comfortable showing principal components plots than trying to explain them. So, as we adopt these different designs, it's important to have some background in what makes them work. When you perform many statistical tests, keep in mind that you must correct for it. When you see a Q value represented in one of these plots, that Q value has already been corrected for using the Benjamin-Hochberg method most typically. So working with Q values might be a little safer to work with rather than the P values themselves. And anytime you're doing untargeted biomarker discovery experiments, keep in mind that you're going to suffer more from multiple comparisons. So if you have a much more concrete set of ions that you actually care about, just the PCs, for example, then it probably makes sense to focus on just those, simply so you'll have fewer tests conducted that might distract you and might force you to call a p-value insignificant when you'd really rather call it significant.